So the end result of mitosis is two daughter cells from one parent cells, and those two daughter cells are genetically identical of the parent cell. So we'll be seeing the mitosis in details. So here is the figure mainly depicting the whole overall process of the mitosis happening. So we know that mitosis happens in the body cells, the somatic cells. So all the body cells, all of our adult cells are mainly somatic cells except the germ cells. So those germ cells mainly undergoes meiosis, producing 4 haploid cells. So there are mainly 5 phases of mitosis: the prophase, prometaphase, metaphase, anaphase, and the telophase. So cytokinesis is actually not part of the cell division, but for the simplicity it is shown here along with the telophase. So, and also note that this prometaphase, actually you will not find always this prometaphase, because sometimes it is shown together with this prophase and prometaphase, but for the simplicity of the understanding it is shown here separated. We will see that. So in the prophase, what happens in the prophase, mainly you can see this centrosome. There is 2 centrosome. Those are mainly replicated earlier in the G2 phase. So there are 2 centrosome, or the centrioles mainly moves to the 2 side of the cell, 2 poles, and the spindle mainly tries to attach to the nuclear membrane. So that's the prophase. But in and prometaphase, we will see that those nuclear envelope mainly breaks down fragments, and those um, and mainly those spindle mainly attached to the kinetochores of each chromosome in the prometaphase. In the metaphase, the homologous chromosomes mainly arrange in line, not side by side. So that is mainly important, the in line. Because in the meiosis the homologs are arranged side by side, but in case of mitosis the homologous chromosomes arrange in line. So this in line is called the metaphase plate, and in the anaphase those homologous chromosomes or sister chromatids mainly separate in the anaphase. And in the telophase we'll be seeing that the cell mainly forms a cleavage furrow, that we'll be seeing that, and mainly elongates in the also cell elongates in the anaphase, but in the telophase the mainly takes the form of like the cleavage furrow. That means the cell mainly tries to separate, and at the cytogenesis the cell mainly divides. So in this figure it is mainly showing the changes, uh, not in details but roughly, in the rough changes are shown here. So moving to the next slide. So the first phase of the mitosis is the prophase. So in the prophase the centrosomes moves at the two poles of the cell, forming the mitotic spindles. So also this well-known characterizing shape of the chromosome is forms in this prophase. So when this condensed or this structure is formed, that is called chromosome. But when they are loosely bound or that means loosely bound and in the scattered form, they are called the chromatins. But when the chromatins are condensed, so supercoiled, forming this structure, then they are mainly referred as the chromosome. So the sister chromatids are like joined together in this prophase, because in the metaphase we will be seeing in the anaphase these two sister chromatids are separated. So also prophase, we will be seeing that in the prophase the nuclear membrane or the nucleolus, those mainly starts to break down. So it's a continuation of the prophase. So in the mitotic spindle, so these spindles are called the astral microtubules or astral spindle. So they have their different function. We'll see that. So these two two centrosomes tries to go end of the cells, I mean two poles. This is the one pole and this is the other pole. That means two sides of the cell. That is necessary for separating the chromosomes equally. We'll be seeing that. So this radial array of microtubules and the side mainly, actually those are short, those mainly the astral microtubules, and at the end of the prophase the nuclear membrane and nucleolus starts to disappear. So in the next phase we will see that those nuclear membrane mainly breaks down fragments. So this is the second phase, it's the prometaphase. So in the prometaphase we see that the nuclear membrane or the nucleus breaks apart, mainly allowing because the chromosome needs to be separated. So if there is a barrier, that means the nuclear envelope is present, then the chromosomes cannot be separated. So those barrier then envelope must be breaks down in this phase. So here are 2 or 3 types of microtubules. Depending on the function of the microtubules, they are named actually. So astral microtubules, we know that what are those mainly attached to the cell membrane, plasma membrane. So astral microtubules mainly attached to the plasma membrane of the cell, and the polar microtubules, from the name, that means they're referring, that means this mainly push the centrosome two sides of the cell. So pole means side actually. That is also referring to the polar. That means this polar microtubules, these microtubules are polar because they are calling polar because they mainly push the centrosomes to the two pole of the cells. And the other microtubule, that is the kinetochore microtubules. If you can see this, that is the chromosome, and at the middle of the chromosome there is centromere, right? So at the centromere there are proteins, the kinetochore proteins. So the kinetochore proteins are attached at the centromere, we know that, right? So some microtubules attach to that kinetochore so that it can pull the chromosome into two parts, right? So those microtubules, those attached to the kinetochore proteins of the chromosome, those are referred as the kinetochore microtubules. So the kinetochores proteins will attach to each of the chromosome at the centromere. So also note that the kinetochore proteins I mentioned, the proteins at first the kinetochores needs to attach to the centromere, and then in those kinetochore at the centromere, then those kinetochore microtubules, and those microtubules then attach to the kinetochore, and those microtubules that attach to the kinetochore are calling here the kinetochore microtubule. So in a nutshell, this is happening. The nuclear membrane breaks down, nucleolus disappear, allowing the chromosomes to be separated, and different types of microtubules uh, are visible depending on their function. So that's the prometaphase. So in the next phase, that is the metaphase.